Imagine a world where we could erase genetic diseases, enhance our physical abilities, and even extend our lifespan. Sounds like science fiction, right? Well, it's closer than you think, thanks to a revolutionary technology called CRISPR. CRISPR, or clustered regularly interspaced short palindromic repeats, is essentially a pair of genetic scissors, capable of precisely cutting and editing DNA. It allows us to rewrite the code of life itself, opening doors to incredible possibilities. But with such power comes immense responsibility. We're not just talking about tweaking a gene here and there. We're talking about altering the very blueprint of humanity. In this video, we'll delve into the fascinating world of CRISPR gene editing, exploring not just its incredible potential, but also the complex ethical dilemmas it presents. We'll unpack the science behind this game-changing technology and examine the challenging questions it raises about our future, our health, and the very definition of what it means to be human. So, buckle up, because we're about to dive into the ethical minefield that is CRISPR. The story of CRISPR is a winding path, paved with decades of scientific curiosity. It didn't just appear overnight. It's the culmination of years of research by brilliant minds building upon each other's discoveries. Our journey begins in 1987, with Japanese scientist Dr. Yoshizumi Ishino, while studying a gene in E. coli bacteria. He stumbled upon a peculiar, repetitive DNA sequence. Little did he know, he'd found the first piece of the CRISPR puzzle. These sequences, clusters of regularly interspaced short palindromic repeats, or CRISPR, were initially a mystery. For years, their function remained unknown. Fast forward to the early 2000s. Scientists began to suspect that these CRISPR sequences played a role in the bacterial immune system. They noticed that the spaces between the repeats often matched DNA from viruses that attacked the bacteria. Then, in 2005, significant breakthroughs came from multiple research groups, including those led by Francisco Mojica in Spain. They independently confirmed that CRISPR sequences help bacteria defend themselves against viruses. Bacteria were using these sequences like a genetic memory to recognize and disable invading viruses. But the game changer arrived in 2012. Jennifer Doudna and Emmanuelle Charpentier, along with their respective teams, revealed the full potential of CRISPR. They demonstrated that a specific CRISPR-associated protein, Cas9, could be used as a programmable DNA cutting enzyme. Suddenly, scientists had a precise tool to edit genes, not just in bacteria, but in virtually any organism. Doudna and Charpentier's groundbreaking work earned them the Nobel Prize in Chemistry in 2020, solidifying CRISPR's place as one of the most significant scientific discoveries of our time. From humble beginnings as a strange sequence in bacteria, CRISPR had transformed into a powerful tool with the potential to revolutionize medicine, agriculture, and our understanding of life itself. CRISPR isn't some futuristic fantasy. It's here and now, transforming fields from medicine to agriculture. In medicine, we're seeing incredible potential in treating genetic diseases. Think of sickle cell anemia, a debilitating blood disorder. Clinical trials are underway using CRISPR to correct the faulty gene responsible, offering the hope of a permanent cure. It's also being explored in the fight against cancer, with researchers using CRISPR to engineer immune cells to more effectively target and destroy tumors. Beyond medicine, CRISPR is revolutionizing agriculture. Scientists are using it to develop crops that are more resistant to pests, diseases, and even climate change. Imagine rice that can thrive in drought conditions or tomatoes that are naturally resistant to viruses, reducing the need for harmful pesticides. This could have a massive impact on food security, particularly in regions facing challenging environmental conditions. But its impact extends even further. Researchers are exploring CRISPR's potential in biofuel production, creating more efficient algae for renewable energy sources. It's even being used in basic research to understand the fundamental mechanisms of life, unlocking new insights into everything from embryonic development to the aging process. The impact on society is profound. We're talking about the potential to eradicate diseases, feed a growing population, and develop sustainable energy sources. It's a technological leap with the power to reshape our world. So, let's wrap things up. We've seen how CRISPR holds incredible potential for treating diseases, enhancing crops, and even eradicating inherited conditions. But, as we've discussed, with this power comes immense responsibility, the potential for unintended consequences, the ethical implications of germline editing, and ensuring equitable access are all crucial considerations we need to address as we move forward. Looking ahead, CRISPR technology is poised to revolutionize medicine and agriculture in ways we can only begin to imagine. We might see personalized therapies tailored to individual genetic makeups becoming commonplace. Imagine a future where genetic diseases are simply a thing of the past, but the key is navigating this future thoughtfully, with open discussions, robust regulations, and a deep respect for the potential impact on humanity. If you found this exploration of CRISPR gene editing and its ethical considerations thought-provoking, be sure to hit that like button. And don't forget to subscribe to our channel for more deep dives into the fascinating world of science and technology.